I was mentored in my research. My research work was mentored by Professor Nalini Adopoli, and my topic is the pure analysis of bioinformatics and phylogenetic results of plant DNA barcode. So, as I'm sure everyone has experienced, the pandemic has warranted some creativity. And when I was actually initially skeptical of how I could conduct scientific research without having access to the traditional methods. So this is actually where bioinformatics comes in. We would be turning the adversity of the situation into opportunity. So Professor Adopoli and myself met over Zoom on a weekly basis, and we utilized the previous wet lab work done by prior Valencia research students, which are the source of my sequences. So this brings me to my thesis. DNA barcodes can simplify the identification and aid in classification of specific genetic sequences for medicinal plant varieties and pharmacology. So what is a DNA barcode? It's a short gene sequence taken from a standardized portion of the genome. Much like the UPC barcodes in retail products, DNA barcodes allows for identification of species with rapid precision. So why is this important? Well, DNA barcoding has a wide array of uses. It can be used in forensics and quality control. It can be used to identify invasive or conserved species. It can identify pathogens. And it also assists in efforts against poaching. New discoveries or novel discoveries are important because Earth's biodiversity is dwindling faster than we can catalog for its species. So what about the phylogenetics? Well, phylogenetics is the study of evolutionary relationships between organisms. And here you can see that there's two phylogenetic trees. And I want to take a moment to note that phylogenetic trees are merely visual predictions of evolutionary relationships. They don't actually indicate where a organism diverged into two species. It represents the relationships between them. So the reason I was bringing this in is because let's say we have a plant that has known medicinal properties. Can we expand research on its relatives? Will they also have medicinal properties? So I came up with a research question. Can we identify a plant to have medicinal benefits from known species in the gene database? My hypothesis was by identifying a plant sequence as well as consulting scientific literature, I would find some benefit. So going to the work of previous Valencia students, you can actually see them in the right images collecting young leaf samples in the field. The Valencia students then took the samples back to the lab in order to extract and isolate a gene from the chloroplast. They then utilized a polymerase chain reaction method to isolate the RBCL gene. So RBCL is ribulose 1,5-bisphosphate carboxylase oxygenase. It is a gene responsible for CO2 fixation in photosynthetic plants, which from the chloroplast. This gene is ideal because of its length for its sequence length, as well as it's the most conserved gene in photosynthetic plants. So after we isolate and amplify this gene, we're going to identify our desired fragment against any unwanted DNA using gel electrophoresis. After we identify the fragment that we want, we're going to send our gene, or they sent their genes to GeneWiz for Sanger sequencing. This brings us to my work. So my work is in bioinformatics. I utilize the DNA Subway, which is a freely accessible web tool. And I prefer it because it actually is an ensemble of bioinformatic tools, and it has a very user-friendly interface. You can actually follow along this line for each series of the steps that we need in order to get our phylogenetic analysis. So you can see here, our first series is to assemble the sequences. We're going to add the sequence to the database. I would like to note here, if it was a novel sequence, we would upload the data and add reference data to that sequence as well. Then in analyzing the sequence, we'd be selecting our sequence and comparing it against other related sequence for the phylogenetic aspect. So I recorded the entire process in a video, and I actually converted each step into small snapshot GIFs. I did this so it would be easier to visualize each step. So I'm not sure if you guys can see it that well, but this part is the sequence viewer. So when we insert a FASTA code, which is a text-based format for each nucleotide in the sequence, it provides two sequences. It provides a forward and a reverse sequence, but the software hasn't identified which is which yet. You may also see in the two sequences, you might notice that there's an N. And this is not a nucleotide itself. It actually codes for an unknown nucleotide or otherwise an unsequenced nucleotide. So moving on from here, the next step is the sequence trimmer. So the purpose of this, it's done automatically for us. We don't need to interfere with this step. What it's doing is, if there's a series of ends or the unsequenced nucleotides at either end, it's just going to trim that off for us automatically. The software already recognizes that it's unnecessary. We're not going to need that. So it just does that in preparation for our next step. The next step is the pair builder. And this is where we're going to distinguish between the forward and the reverse strands. Now, this is important because when it comes to DNA strands, they're anti-parallel to each other. We need to indicate that so that the nucleotides are complementary to each other. So let's say that this is the 5' end of a strand, and this is the 3' end of the strand. We want it to be anti-parallel to each other. If the strands are parallel to each other, it is not going to be the same sequence. The nucleotides are not going to be complementary to each other. And the DNA strand, from this point, the sequence would be corrupted because it's not indicative of the actual plant DNA. So after we have indicated which is the forward and the reverse, which I'm not sure if you can see here, this one has the file name 25-M13F. The F indicates the forward strand. And much like that, the 25M13R indicates the reverse strand. So I know that I'm going to change that F to an R, and I'm going to select both to automatically pair together, which is in preparation of our next step. So now we're in the consensus editor. We can now see 3 strands, and this 3 strands, the top 2 are the forward and reverse sequences that we have been seeing previously. The consensus strand is actually based off of the forward strand. The goal here is to actually snip or trim the undesirable ends of the sequence. Because as you can see, from the beginning, there's like a series of dashes as well as yellow blocks. It's also the same case for the end. So dashes actually are like placeholders. So if, let's say, the forward sequence has a series of nucleotides, yet the reverse sequence is missing the unsequenced nucleotides in that same location, it'll just put the dashes there because if the nucleotides are missing, it's going to malalign. So we want to prevent that. The yellow blocks are actually mismatched nucleotides. So let's say, for instance, we know that A pairs with T, but if the 2 strands are indicating that A is with G, it's going to know that there's something wrong, but it can't determine which one is the incorrect nucleotide. So it's going to show that in the yellow box. So what I can do manually here is I can trim the consensus. So I can trim either from the left end going right or I can trim from the right end going left, because we can't trim from the middle. It has to be a sequence. It can't just be, random blocks of nucleotides. It has to be one solid sequence. So after this is the exciting part. I can take this sequence and I can run it through BLAST. And BLAST stands for the Basic Local Alignment Search Tool. And you might notice that it says BLAST N. The N part of the acronym is actually for nucleotide, because there's also a database search tool for proteins. Now, BLAST, it works like a Google search for genes. We're going to tell it information, and it's going to pull up the most likely match to what we're requesting. In this case, we finally can put a face to our sequence, and it is Catharanthus roseus. So after this, I did some research into, now that I had the genus and the species name, I did some research to find the common name as well as its appearance. And here's a funny story. When I learned the appearance of this plant, it had a striking similarity to a plant in my backyard I tried to kill not 2 weeks prior to discovering it, because it was overgrowing the sidewalk. I was frustrated. I just hacked it apart. It survived, but it's just funny. I didn't realize that I would need it later. So I identified the common name, which is the Madagascar periwinkle. And I utilized the Picture This iOS application. And how this application works is it essentially confirms a plant based on its morphology or appearance. So it confirmed that it was in fact the Madagascar periwinkle by appearance. I also learned that although this is used for ornamental purposes generally, it also is known for its medicinal purposes. It's very exciting. So I consulted numerous scientific articles regarding the benefits of this. And it was a bit overwhelming. So different parts of the plants are going to contain bioactive components, usually in the form of alkaloids. So in the root specifically, it has the alkaloid agemalacine, which works as a hypotensive, and that can be used to reduce arterial blood pressure for hypertension or even anxiety. In the flower has wound healing capabilities. There was a study that was tested on rats, and it was shown that the component in the flowers increases epithelial tissue healing as well as wound contraction. In the leaves, as a disclaimer, this plant is very commonly used in traditional medicine. The juice from the leaves increases insulin production from the beta cells in the pancreas. One study demonstrated that in countries where medicine isn't as accessible, people will chew the leaves to help control their diabetes. The leaves also have a hypolipidemic effect, especially against high LDL cholesterol levels. It also has an antimicrobial agent, especially effective against gram-positive bacteria like Staphylococcus. Found in the whole plant, another exciting one. In the whole plant are two alkaloids that may be recognizable, vincristine and vinblastine, which are used in chemotherapy to inhibit cancer cell division. Also found in the whole plant are antioxidants, which can be used for inflammation or delaying cell damage. So I discovered that this plant has a plethora of medicinal purposes. So what I want to do now is I want to go back to the DNA subway. And this step, I'm selecting data, but this data actually originates, we're going back to the BLAST. The BLAST not only pulls up my species, but it also pulls up any related species that might have similar fragments in its sequences. So I select I would say 23 separate species based on their genus or species name. I wanted them to be unique. And I select all of them for the next step. So at this point, I'm utilizing the muscle tool. Now, muscle stands for multiple sequence comparison by log expectation. And it's a really wordy way to say that it aligns up to 500 sequences within seconds. So it uses a comparison point between these sequences. And better seen here, these are the results of the muscle tool. So it's showing these blocks are actually indicative of like color-coded nucleotides for better visualization. And the alignment viewer itself is just showing the similarities and differences in certain nucleotides within the sequence. At the bottom, you may have seen it in several of the borders I used. In the bottom is actually the DNA barcode of my species, and it's beautiful. So what can we do with this data? So the DNA subway provides two types of phylogenetic trees. For the sake of this research, we're going to just use the phylogenetic neighbor joining tree, but there's also the maximum likelihood tree for reference. So I take that data and then I'm going to select the phylogenetic neighbor joining tree, and to the right are my results. This is where my species is. And the purpose of this is I wanted to see if my plant had medicinal benefits. I wanted to see if maybe its genetic relatives may also have medicinal benefits. So this brings us to the other species. So here is Parahencornia fasciculata. This has a latex used in traditional medicine for a treatment for yaws, which is a contagious disease caused by a bacteria, and it can eventually lead to skin ulcers and lesions. It also is used to treat other external ulcers as well as other skin disorders, and it can be commercially processed to treat internal ulcers. Another relative is the Lachmelia aculeata, which is used as an anti-diarrheal and a febrifuge, or otherwise known as a fever reducer. So what does this mean? Well, it may not have the same medicinal properties as the original species of my sequence. However, I actually ran into an issue where I could see that there were traditional medicinal values for these plants, but there wasn't much scientific literature going into the pharmacological aspect. Can we use these plants that we know are traditionally beneficial and process them for pharmaceuticals? So I feel like a phylogenetic tree can serve as a jumping point for more thorough research of these species. So in conclusion, using DNA barcoding, we can expand future research on the pharmacological applications of samples, starting with identification for a sample's unique bioactive components, as I did here. Our development of phylogenetic trees and connecting beneficial samples with its relatives opens the door to expanding our knowledge on medicinal plants. I would like to take a moment to thank Valencia College, Seneff Honors College, my mentor, Professor Nalini Adopoli, Dean of Science, Dr. Robert Gesner, Professor Dr. Melanie Sexton, Professor Jane McGuire, Advisor Kara Coyer, and the two software applications I use for my bioinformatics, Biorad and Cyverse DNA Subway. Here are my references. They're derived from medical and pharmaceutical articles, as well as information for bioinformatics and phylogenetic trees. If you have any questions, I would be more than happy to answer them.
I have a comment. That was very well explained, and you did such a great job that even I can understand. So good job, Sophia.
Thank you. I really
I
tried to, oh, go ahead.
Oh, I just wanted to say I agree with Jane. I mean, seeing a more expanded, detailed version of it this time, I didn't realize how much I missed the first time, that I saw part of this. And I really needed to see it a second time, but it was well explained because I think I get it actually. And actually, I mean, in general terms, but honestly, the first time I saw it, I thought, oh, this is a hard subject. I'm really going to have to pay attention here. But I really, the way you explained it, I could follow it. So I appreciate that. I'm a little surprised, I have to say, but pleasantly surprised. You did a great job.
Thank you so much.
And
Sophia,
Sophia,
go
me
on.
not having a science background at all, I can say I understood you. Your explanation was really clear, and even though it's very scientific in nature, it was simplistic in a way that it needed to be. And I think your message overall was very well conveyed. Very good, really good.
Thank you.
Sophia, you did an outstanding job. This is phenomenal. I wouldn't even need to read your research to understand what you were explaining. I think the explanations are very clear. Could you talk about, you talked about PCR and gel electrophoresis to separate out the gene of interest and the sequence of interest. When you do PCR, what exactly happens there? And I don't mean
So,
the enzymes. I mean, what's the general idea to do PCR?
so we're going to have some isolated DNA, and we want to target specifically our gene of interest. So we're in PCR, we're going to include the primers that specifically target the coded ends.
Mhm.
So that way we know this is our RBCL gene, this is the one we want, and then we're going to amplify that to be used.
Good.
Did I answer your question?
Yes, you did. Yeah,
Okay.
I'm just looking at some of the background information because I'm so impressed. I can't explain how
Thank you.
impressed I really am. Are you still at Valencia? Or are you a USF or UCF?
No, I'm still at Valencia. I'm actually majoring in nurse. Well, I'm doing both. I'm majoring in nursing. I'm actually going to enter the nursing program this summer, or register for it. And I'm also trying to get my AA because I'm kind of torn. Like, I have fallen in love with science, so it is killing me to decide where I want to go in that future. Like, I love biomedical science.
Mhm.
The reason I tried to like bring this down, because in the conference I actually used a script. I actually wanted to bring this down because I feel like I wasn't conveying like how exciting this research is, how exciting DNA is, how exciting even coding is, which is notorious for not being exciting. It's so I just wanted to try to highlight those things.
Your
But
code,
yeah, oh, go ahead.
your code is beautiful. I agree. You should actually have that printed on something, make clothing out of it.
Yeah, it's
I like the DNA subway idea. I could have sworn I was on that subway in New York City. And I mean, the whole thing was so well done. How do you see, well, I think you're starting to answer the question. My impression, being impressed is at one level. Knowing that you're still at Valencia, it's just gone to a totally another level. Because after being at Valencia and not having gone on further for further education, I'm Professor Adopoli. I already know what she can do, but Professor Adopoli, you have done wonders with Sophia.
Thank you.
This is phenomenal. What do you think you could do in the future? Because nursing is not a research-based kind of degree, and so you may eventually go into a doctoral program where you combine these two things because they're very different in many ways, but it'd be very interesting to see how you could actually combine these in the future. Where do you think, one of the questions I had was, where do you see the use of your research in the future? For instance, if you could identify these pharmacological agents and extract them, do you think that you could do research, for instance, to have them produced through gene amplification and like they make insulin in bacteria?
I'm actually really excited you asked that. So one of the prospective things I wanted to continue. So how I mentioned earlier that countries like the Philippines, which is my mom's home country, that's why it was significant to me. Sometimes they do not have the same access to medical treatments. So if these plants provide properties that can even be used in its traditional medicine form, where it doesn't have to be extracted, it doesn't have to be processed. Like, can we cultivate these plants to help places that don't have the access to pharmaceuticals?
Sure.
Can we provide healthcare for the world without the same, with the resources that we can provide them?
Yeah.
For nursing, the difficulty is, like, I would love to expand on the pharmacological aspect of, plants and DNA. How can we help these people? And like I mentioned earlier, with the lack of research on the other plants, is what are we missing? What can we do more to help more people have the medical care that they desperately need? The yaws, it could have been entirely preventable, but they found a way to treat it despite having no resources. It's completely impressive. The more I delve into it, the more I'm excited. And I just, my goal is to really convey that excitement to you guys. So
imagine if you could take the gene that produces the pharmacological agent and genetically engineer the plants that are commonly cultivated in these different societies. If they orally eat the foods, the leaves, the fruit, and they get treated that way. I mean, there's also,
yeah, it's incredible.
yeah, this you did a great job. I cannot express how and really happy I was. As everybody said, this is so well organized, so well presented. You were so confident in your pronunciations and your understanding of the concepts. Everything was perfect.
Thank you so much. It means the world to me.
Very.
I'd just like to, I know Bob kind of asked some of my questions, so I just want to make a quick comment. Sophia, you were in my class last semester, and you and I had a lot of heart-to-hearts, and, just in terms of, what is your major? What is your research area? You were looking at psychology, nursing, into biology. So I know entering the fall, doing a research methods course online, there was a lot of confusion, frustration, and, me and you, we worked together. And I just want to say, I'm really, really impressed with your growth. I don't think everybody has that same backstory, but to see you go from, confused, not sure what research is, to this phenomenal, you're a biomedical researcher. That's what it stems to, in just a short matter of time. It's very impressive, and I'm very, very proud of the work that you've done here. So, take that with you. I know we were struggling together, but it worked out for the best. I would hope you would agree. And so
I do.
I just wanted to know, kind of like what Bob said, what is the next steps? Like I said, you're kind of in all these interdisciplinary areas. I am curious to see what your future plan is, and if you can create a path that does involve research to some extent, because I think you have a lot to contribute.
It's so, I'm really at an impasse right now. I would really love to thank my mentor, Professor Adopley, because my love from science actually stems from her. As she said, I was in her biology class, I was in her microbiology class, and I'm in her research class. All of my love from science comes from her. She supported me all the way. And I do like that you mentioned, right before I decided to do the scientific research with her, when I was in your course, I was preparing for psychology because I was sure that I was going to be a psychiatric nurse. I was sure. So when the mentorship fell through, I consulted Professor Adopley, because me and her, like, I know that she's going to bat for me, and I know that she knows I'm going to put in the work. She tells me to do it, I will do it. So I asked her, and she said, we're going to do DNA research. And in the aspect of the pandemic, like I said, I was skeptical. Like, how are we going to do that outside of a lab, Professor? But no, she has guided me throughout all of this. I would like to note that she has taught me the phylogenetics, the bioinformatics. All of this, I believe, is actually taught in Bio 2, is it not?
Yes.
I did not take Bio 2. All this information I had to learn, and from her, and she was so helpful in that. But when I'm sorry, I got off track of your question. In the future, I really think that I'm starting to want to go more towards biomedical research. I love research, and that's from you too. And Angela, I love research. So I want to keep doing that. And I know nursing is not going to do it, at least not until the doctorate level, which I'm still aiming for, but I'm still thinking about it. I got to find out how I can do this.
Yeah, I realized that if you were going to nursing school, have you been accepted?
I haven't registered yet.
Okay. If you go to nursing school, you'll certainly be very successful there too, because your dedication is something that's going to make sure that you are successful no matter where you go. Whatever you choose to do, you have shown the dedication that you need in order to be successful. And I had a feeling you hadn't taken bio 2 because nursing students don't take bio 2. And that's why I started to get, I'm thinking to myself when you said that, I'm thinking, wow. Because a lot of, I had to say, and I already said it in the chat, I know what Professor Dompoli can do, but Professor Dompoli and Nalini, you did an awesome job. Just awesome. I already see what she's done with other students. This is like another level.
Yeah, this is research. So I try to go up with the standard, and yeah, I had to take the rigor to the next level, the research level. May I take a few moments here to congratulate Sophia as well as ask my question? Is that okay? Anybody else has any questions? Yeah. Wonderful. First of all, thank you very much for being so cooperative, Jane, Melanie, throughout the process from the beginning of the inception of this research course, honors course, and Angela, John, and Bob for joining us and trying to understand what this is all about and giving all ears and all your attention. That was phenomenal as well. And coming to Sophia's work, as I said, she was with me from 1 year now, right? And she has been a great student. I call her stellar performer, but today I would like to give her one more title. Sophia, you deserve this. You're a stellar scientist. You are a scientist today, by all means. Yeah, you are a scientist. You know that. Well, I'm looking forward to you come join me in the lab, and we do the wet lab research as soon as the lab's open. And here is my question for you. I bet you can give us a little glimpse of what's going on there. I never see GIFs or GIFs. Why did you even make those GIFs? And just walk us through what was all that about and how did you do that? So some background, I've actually, I've been working on computers and software since I was, very young. I understand how boring coding can be. I wanted a visual walkthrough because coding, it's really hard, just in general, not just with the DNA coding, it's really hard to follow along the steps. And I didn't want to fall asleep for my own presentation. So I actually use a screen recording for the entire process through the DNA subway from the moment I insert the file to the very end. And I wanted, instead of playing a whole video, I wanted to be able to walk you through the steps, take my time. I wanted you to repeat. So in case you missed one of the steps, you'd be able to see it. So that's why each slide has its own GIF, because it's important. Let's take, for instance, the consensus editor. It's actually, let me see if I can go back to it for just a moment. The consensus editor, nope. So the consensus editor, if I would have just taken a screenshot here, I feel like you would have either missed how lengthy the middle sequence is, or that there's also unfavorable data at the other end that we need to trim. We're not just trimming one end, and that's why we have to trim going in. So without visual depiction of this process, I feel like it might have been harder to explain, or at least to maintain visual interest. So that's why I did that. I hope that helped in this. It was very helpful because we could see, and it was repetitive. So as you were explaining, you were able to point out. So that was very impressive. Maybe I should start using some GIFs in biology and microbiology. Well, how does endotoxin come out of E. coli 015787? Sorry. And that's completely true. The concepts, the scientific concepts would flow together more easily if I understood how they all work together. Even just one concept, if you see how that was given rise to, it makes so much more sense because it's all pattern, right? Yeah.
And how many hours did we spend, if you like to share with the panel?
I don't know how to really put a number on it. It was a lot of hours.
Exactly.
I honestly do not have the most favorable relationship with my computer right now. I can't wait to take a break from it. Honestly.
Yeah, so that was scientist Sophia Honeycutt giving us a little preview on her first research ever. Looking forward to more and more of these. And thank you, everyone, and happy rest of the day. Enjoy.
Thank you.
Thank you.
So that was an
scientist.
exceptional presentation, Sophia. I have a question for you. Do you have a message for future scientists or DNA barcoding researchers at Valencia?
My message to future scientists and researchers would be to find your love and your passion for science. It is going to guide you through all of the sleepless nights, all of the adversity, all of the incredible, grueling data for analyzing. That love is, that love is intoxicating.
Truly, it is addicting, right? Yeah, yeah. And we can do so much more with this research. Yeah. Okay. You want to add anything else? Wonderful. Thank you and have a great day. That was a wonderful presentation.
The scientific research process, it can be very discouraging. So you got to find that love for science, because it's going to keep you pushing. Every new discovery is going to inspire you to go even further than you thought you were going to. I went from wanting to do psychology to now reconsidering my entire future because I love scientific research. And if you find that one thing that you'll fall in love with, it's going to take you so far.
Wonderful.